Hello. Thank you, everyone, for being here with us today. We really appreciate everyone who made it out here, both online and in person. My name's Naomi Martin. I am an assistant business editor at the Boston Globe. I have the privilege of overseeing our biotech and healthcare business reporters. And it is my absolute pleasure to be here today with Dr. Priya Chakalingam, VP, Head of Clinical Bioanalytics and Translational Sciences at Beam Therapeutics, and Dr. Christine Duncan, Medical Director of Clinical Research and Clinical Development of the Gene Therapy Program at Boston Children's Hospital. Thank you both so much for being here. So today we're going to be talking about pioneering breakthroughs in gene therapy. And I wanted to just, because you both obviously are so in the world of this, give you a chance to just for a few minutes, give people a little bit of the highlights of what kind of groundbreaking work you've been involved with in this space, what kinds of rare conditions you work on, and what do these developments mean for patients' lives. So I don't know, I guess Dr. Duncan, do you want to start?
I'm happy to start. So first, I do want to thank you, Naomi. Thank the Boston Globe, Alexian, and all the sponsors for this event. It's truly remarkable, and I'm so happy to be here. My path to gene therapy started in 2011 when I started working with Boston Children's and Bluebird Bio and companies that were developing gene therapies for adrenoleukodystrophy. I am trained as a hematologist and an oncologist with a focus in stem cell transplant. And I think I was telling some people earlier, if you told me that my work was going to switch completely from cancer to rare disease, non-malignant disease, I'd think you're crazy. And I'm so glad that I had that opportunity to make that switch. So I consider myself very lucky that I got to work in the development of clinical gene therapy for adrenoleukodystrophy, mostly because I had the amazing privilege to work with the families of our patients and the advocates, and that has been life-changing for me, not just career-changing, but inspiring. And so now I no longer actually treat oncology patients. All I do are gene therapies and cellular therapies for children who have rare non-malignant disorders. I was very lucky that I got to enroll the very first patient in the world on in the clinical trial for gene therapy for ALD, and then also 10 years later, was able to be the physician who treated the first patient on the commercial product. And so most of my work is in adrenoleukodystrophy, but I also work with metachromatic leukodystrophy and in the mucopolysaccharidosis space, and osteopetrosis and other rare similar diseases.
Awesome, thank you. And Priya, why don't you talk about what you've been involved with?
Sure. Thank you, Naomi, Boston Globe, Alexian, and all the participants for this great opportunity, and thanks for listening to us. My basic training, biochemistry, and my connection to rare diseases started with my postdoctoral work at the University of Tennessee Medical School more than 25 years ago. I worked on Duchenne muscular dystrophy to begin with. And after that, I've been with Wyeth, which became a part of Pfizer, then Shire. As you all know, it's a big rare disease company, Shire, which later became part of Takeda, then Biogen, and now I am with the Beam for the last 4 years or so. I lead a function called Clinical Bioanalytics and Translational Sciences, reporting to the CMO. So at my work on gene and cell therapy started when I was at Shire. We worked on a lot of, as you know, lysosomal storage diseases, hereditary angioedema, all major drugs in the market, they all started, not all, most of them started at Shire. And then Biogen, neurodegenerative diseases. And at Beam, Beam leverages base editing technologies combined with cell therapy, like allo or autologous cell therapy and CAR-T therapy too, combined with novel drug delivery modalities like LNPs. So that's basically what we do. And currently we work on hematology, primarily sickle cell disease. And we have a clinical study ongoing, Beacon study to treat sickle cell patients. So it's actually a base-edited, autologous, base-edited HSPCs given back to the patients to treat. And it's ongoing actively. We presented data at the ASH. And we also have programs on liver So it's in vivo editing, where we deliver the cargo through LNPs. So we have programs in AATD, alpha antitrypsin deficiency disease, and also glycogen storage disease type 1. And then we have others coming up, PKU, and so but these are the major ones.
Amazing, thank you. So I just wanted to give a shout out to this Boston Globe story. If you guys want to Google it, you can search for Boston Globe, and then the name is CONNERHESS. So it's Connor with an E, Hess. This is a 6-year-old boy, he was 6 2 years ago, who Dr. Duncan treated at Boston Children's Hospital. And one of my reporters, Jonathan Saltzman, wrote about it. And it's just an incredibly moving story. This treatment that he received, which is marketed as the name Skysona, cost $3 million, but is a life-changing gene therapy for cerebral, I'm not even going to try to pronounce it, ALD, cerebral ALD. This devastating disorder strikes boys, usually between the ages of 4 and 10, causing rapid loss of cognitive and physical abilities, including hearing, vision, and movement, often only months after the first symptoms appear. Most boys die within a few years. So, Dr. Duncan, you were involved with giving him this life-changing therapy. Can you talk about how he's doing 2 years later and what it feels like for you as a physician to see these advances come to life in your patients?
Absolutely. So the timing is perfect. And I do want to thank Jonathan Saltzman for working with the family. It was an incredible experience. So actually, Connor's birthday was yesterday. And so I don't think he's watching because he's in school doing perfectly well. He's in first grade. But I do think his mama is watching. So thank you, Rochelle, if you are watching on Zoom, for giving us the opportunity to be part of that journey. I think for all of us who work clinically and not clinically, sort of in the lab, everywhere in the rare disease space, it is not just a job. It is not something you don't check in, you don't check out. For many of us, all of you in the room with rare disease. But Connor's journey and so many of boys like him and families like them is sort of why we get up in the morning and why we do our job and why in uncertain times we continue to do this. It's because of people, children, adults, everyone with rare diseases. And it's really, I say it's a little bit selfish. Like, I get to be part of that, and I do not take that for granted for a moment. And so incredibly grateful and incredibly happy to see him like smiling and goofy on his birthday. And that is because of the work from patients, families, advocates, industry, academics, all of us coming together. So it's incredibly meaningful, I think, for all of us, and to see that. So please do look at the story and just know that Connor is always sleepy at medical appointments, so he looks very sleepy now, and the pictures on that story, but he is up and playful and running around. It's great.
It's so amazing. And he has like a relatively normal life from what you know.
He has an absolutely normal life.
That's incredible. So Priya, I know you've been involved with biomarker-driven approaches. I'm wondering if you can give a quick description of what that means, and then also talk about how can they help with expedited approvals, especially for rare diseases?
Sure, yeah. So biomarkers, they're very close to me, like translational medicine, translational sciences. That's my background. So we do not only strategy, but also biomarker discovery, development, and implementation and data generation. So for rare diseases, biomarkers can help in the accelerated approval of rare diseases. So some examples, like for PKU, it has been accepted with that blood levels, that was used for accelerated approval of a drug, blood levels of phenylalanine. And then another example is for Duchenne muscular dystrophy. You could use this microdystrophin protein expression levels as a biomarker, and that was used for accelerated approval of a drug. So in our case, like I talked about AATD, which is alpha antitrypsin deficiency disease. So there too, because this disease happens, it affects the patient's both liver and lung. And this disease is caused by the SERPIN1 gene. So there is less of this protein AAT, alpha antitrypsin. Blood levels, like functional protein levels, are low. But whereas the toxin protein, the ZAAT, accumulates in the liver, causing the disease. So the blood AAT, so we are working on several biomarkers, like a blood AAT measurement, functional AAT, then ZAAT, there's Z polymers. So many of these could actually help in the accelerated approval of this drug. So that's one example where the biomarkers can help in the accelerated approval of drugs. The other thing is, like, we work on sickle cell disease. So we use the VOCs, the vaso-occlusive events, right? But that takes time, and you just follow patients for a long, long time. But then are there other ways to see if our gene cell therapy is effective? There are ways we could look at the function and the health and function of the red blood cell itself. So these are all exploratory biomarkers, but they help in just telling us a story that yes, there is resolution of sickle cell SCD pathophysiology. So we could look at how sticky the blood is, the rheology of it, like the stickiness, the deformability, their ability to deform and get through the vessels, and their adhesion properties. And then actually recently JHU even showed that you could do in vivo rheology, because until now we were taking patient blood and doing ex vivo rheology, but they've come up with a very novel way of looking at in vivo rheology using tongue. So all these are biomarkers which can tell, even earlier, even before all these VOCs, we can tell, oh, the blood health is improving, the function is improving. So that's how biomarkers can help, especially in these rare diseases, because we need something, to tell us that things are working and the patients are getting better. And
Thank you so much. Along those lines, obviously we want to know when things are working. Why is it so important to follow patients for a long time in the rare disease space? And also, what are the challenges with following them sometimes as much as 15 years?
So there are some really important reasons that we follow people, just speaking specifically about gene therapy. So the FDA mandates for gene therapy, the ex vivo type, where you take a patient's own blood stem cells, they go to a laboratory, a healthy gene or a change is placed back in, and then those patients get the cells back after chemotherapy. So that's this one path. And the FDA requires 15 years of follow-up. So there's the mandatory piece, but there are also the really important pieces, which, one, we need to know that the commitments, the time, the efforts that people are putting into this, the patients especially, are worth it. Are they working? What is the quality of life for a patient who's gotten these therapies? And so I think that is the really important thing, because that helps to drive the next innovation, that helps us figure out what we need to do. But then the other critical element is safety. And so I think about our journey with adrenoleukodystrophy and gene therapy as one of sort of highs and lows. And one of the lows that we've experienced, and I know many people in the room are probably aware of this, is that despite all of the best preclinical safety information, all of the testing, everything that we knew at the time, there are boys who received out of 67 on a trial and now more commercial, there are boys who've developed blood cancers as a result of that. And if we are not following people once drugs or products are commercially available, we are not going to know how to improve on that. We're not going to know those things. So it is the ups and downs of technology and moving forward. And so that's an incredibly painful reason that we need to follow. But then in the in vivo space, which is different, where you don't get chemotherapy, you don't donate your stem cells in that same way, where a stem cell or when a gene therapy product is given to a patient, either injected into their eyes or to their spinal fluid, or in cystic fibrosis even inhaled, the requirement to follow is less, usually around 5 years, but it doesn't mean we shouldn't follow those patients for the same reasons. We want to know how we are doing, how those therapies are performing, and how we can do it better. And I think one opportunity that we have is for all of us in the rare disease community and the gene therapy community to come together so that we're not looking at the safety for one sickle cell project or product compared to another, but all of us coming together so we can find out in the short term how to do this safely, but also in the long term how to make sure that patients are getting the effects that they need and that they deserve.
And I know when we spoke before, you mentioned that you have a lot of patients from all over the world, right? And from the country. How hard is it to follow them, and who's doing it?
Yeah, it's incredibly hard. It's a great question. So on clinical trials, especially, there are patients really who are coming from all over the globe for therapy. And then they're in the commercial space, there are as well. And so once a patient goes back home, I recently had a patient come for ALD gene therapy from Europe, it is very difficult to try to follow. And so I think it's key, and we heard this from the other speakers, that we partner with people where they are from and where they are at home to try to figure out how we can make those connections, and that we also stay in contact with them. So for products that are in a clinical trial, it typically falls to the sponsor, whether that's academic or industry, to follow long term. Commercial is different. The FDA approvals will require some follow-up for some products, which may be different for another product. Some industries have been fantastic about trying to develop that. Some of the follow-up is coming from patient advocacy groups and some from academics. So I think one of the things that we need to probably tighten up or optimize, if you will, is how we are following patients across different platforms globally, across the United States. It's just so that we can make sure that everyone is getting followed and getting followed closely.
Thank you. Priya, I'm wondering if you can talk a little bit about, how we've seen so much clinical promise with these gene editing and gene therapies, but then we've also seen so many biotech companies that are working on this struggling financially, and that could affect these therapies' future availability. Why is it so difficult to make these treatments into commercial success, or is that not the problem? Or I'm curious what you think of this.
Yeah, it is a challenge. The reason is there's so many things. First of all, there's gene and cell therapies are very expensive. They're costly, right? And the cost, why is it so expensive? Because, the manufacturing process, it's very extensive, requiring very specialized facilities and regulated facilities. And then even the entire process, if it is like, for example, the sickle cell disease, it's autologous ex vivo. So we have to take the cells out of the patient and, culture them and do the editing, the part of gene editing and all that. And then given back to the patient, and the patient has to be admitted in the hospital until we make sure, until the engraftment happens, and that's a few days. So the entire thing, it's just things are expensive. And then the other thing that adds to it is we are working on rare diseases that have very small patient population, right? So there are only so many patients you could go after. And then the other thing is, like, the companies also have to present this expensive one-time treatment that's kind of to make it accessible to both the patients and the payers. And so these are some of the challenges that we are facing. Why it's just commercially challenging. And so, and it's evolving, right? We are all the companies working on gene and cell therapies, we are all learning together, and things will improve, and we are going from ex vivo editing to in vivo editing and making it much more safer. So over time, I guess, things will evolve, and we'll find ways to scale up the manufacturing and make it cheaper and more affordable. And also when it becomes in vivo, probably, that might help. So these are some of the challenges, like the cost and the small patient population. And these are the challenges that we are facing. And then added to, like what Christine was saying, like the 15-year follow-up, to make sure, it's safe and efficacious. Like we do editing, base editing, we have to follow making sure the PD effect is still there, the editing is happening, but there is no off-target editing, and there's no insertional mutagenesis or anything, and as she pointed out. So all these things kind of add up to, the challenges.
Okay, thank you. And obviously, the federal government has a huge role to play in kind of stepping into some of these, challenges that the private market has a struggle with. But we've also seen so many changes happening lately on that front. I'm wondering, Christine, can you talk a little bit about what are some of the changes and cuts in how those could potentially affect people with rare diseases that you see?
Absolutely, and I think we've heard a lot about the NIH cuts in research today, and that's incredibly important. I also want us to think about some of the other areas and the other programs that are being affected. So as a pediatrician, I think a lot about early intervention. I think a lot about Head Start. I think a lot about Medicaid and how children get coverage. And all three of those programs, are being proposed for massive cuts. And how does that affect a rare disease patient? So just by the diagnosis in pediatrics, almost, depending on your state, but almost every state all of those diseases qualify for getting a child early intervention. So from 0 to 3 years old, services that come to your home, physical therapy, occupational therapy, all of those things that we rely on for excellent patient care and optimizing that, but we also rely on for clinical trials and outcomes. It's critically important to children, and children are going to be left behind if that is cut. Head Start, the same thing, which does early child education. People will be left behind. And we can't say enough about Medicaid. And so I think we did hear earlier today about the work piece and all of those things. It's incredibly important. So absolutely, we all need to sort of get together and fight the research cuts, but we also need to think about those real tangible things that are coming to a child in their home to make sure, whether it's a leukodystrophy, muscular dystrophy, any of the other diseases that can be affecting children, that kids are getting the services that they need, because they are certainly not guaranteed. And we used to think that those were untouchable programs, and I think we're learning that everything is touchable right now.
Thank you. All right, so I want to talk about what's next in the rare disease treatment space. I know you mentioned there's sort of this shift happening between ex vivo and in vivo. Can you guys both, I guess, Priya first, talk about what you see as being next and that shift?
Sure. So, like I talked about sickle cell disease and liver programs at Beam. So the difference is sickle cell disease, it's ex vivo, and then whereas the liver, it's in vivo. So where we deliver the entire gene editing cargo into the patient through LNP lipid nanoparticles. So the cost is a major factor, because you can actually, when we do the in vivo editing, we can control the cost a lot. And imagine if we were to convert the sickle cell thing. We are working on, even for sickle cell disease, we are working on in vivo editing. And you can cut down all this, the patient being in the hospital for the engraftment to happen. All those things can be cut. And if we are able to come up with the safe in vivo editing tools, we can cut down the cost significantly. So like, imagine countries where only one-time treatment visit is possible for a patient, like they cannot even think about this ex vivo right now, right? So it'll be a game changer for countries where, the patient visits are limited, or their affordability, So this in vivo editing will be a much more affordable option for countries, that are limited.
Absolutely. I think that is incredibly important, bringing therapies to people, places wherever you are that need it. I think from the science, we're going to hear this afternoon from Dr. Tim Yu and others about sort of the next steps. So starting with this ex vivo, we're putting a gene in and giving people chemotherapy. And as we funnel down to how to do this more safely, I think the other things to think about, as I said, I'm an oncologist. I don't want to give another patient who doesn't have cancer chemotherapy. I think so next steps, we need to figure out how, while we're still giving ex vivo therapy, to do that safely without chemotherapy. What's next there? So I think that is critically important for us to do. I think it's also important that we think creatively about how to bring these therapies and the follow-up to people. I often think about, there's this van, it's an ophthalmology van that sits outside our hospital every so often. You can go out there, get your vision check, get your glasses, and move along. And that's incredible. But we need to think about that beyond sort of the therapies. Like, I think we do that for blood donation and other things. But can we use that model to go and collect stem cells from someone who lives in a place that doesn't have a stem cell apheresis and then bring that to the center? So I think the science is incredible, but there are some on-the-ground things that we can do that might bring these therapies to people rather than making people come to us.
Thank you so much. All right, I think that's it for our time, but we really appreciate you all listening and appreciate our guests so much. And yeah, that was really enlightening. Again, my guests were Priya Chakalingham, VP, Head of Clinical Bioanalytics and Translational Sciences at Beam Therapeutics, and Dr. Christine Duncan, Medical Director of Clinical Research and Clinical Development of the Gene Therapy Program at Boston Children's Hospital. So thank you both so much.
Thank you. Thanks. Thank you.
Thank you to the Boston Globe and all of our panelists who have given us insightful programming today. My name is Kristen DeAndrade, and I am a woman living with achondroplasia, a form of dwarfism or skeletal dysplasia. Growing up, I faced many of the difficulties that have been spoken about today, whether it be diagnosis or treatment options. And ultimately, my parents wanted to give all options available to me, which ended up in me being excluded from my community, the dwarfism community. Thus, I have gone forward and formed Little Legs Big Heart Foundation, which is a nonprofit we support individuals with skeletal dysplasia to live life beyond height. I know firsthand how tough of a journey it can be and the complexities of rare conditions, not just for the people who are living with them, but for the caretakers, the families, and the teams that join us on our journey. After lunch, I will be moderating the panel, Making Space for Tough Conversations in Achondroplasia, sponsored by BioMarin, whose work translates the promise of genetic discovery to address genetically defined conditions. We're going to talk about our experiences and complexities openly, honestly, and with the voices of those who live it every day. We have an incredible group of panelists who will share their perspectives, from a parent making difficult decisions for her child to doctors guiding families through this journey. We know these conversations are not easy, but they are incredibly necessary. Maybe you aren't directly impacted by achondroplasia, but I bet you're here to learn, and I invite you to listen, reflect, and most importantly, engage in this conversation. Hope to see you.